Thank you. Reem, do you want to go ahead and introduce the patient expert and the clinical experts at this point?
Sure, I'm happy to do that. So today we actually have two clinical experts, and we're going to be joined by, I believe, one patient expert. Why don't we go ahead? I'm going to start with the clinical experts. Please go ahead and introduce yourself. Oh, maybe the patient expert, sorry.
Yeah.
Kimberly Martinez, go ahead.
Hello, my name is Kimberly Martinez. I'm a NASH patient and a liver transplant recipient, post-transplant 7 years. And I'm very honored to be here. I'm so excited to hear talk about a new drug for NASH, or a drug for NASH, since there are no existing drugs. Thank you so much. And I have no update to my statement of conflict.
Welcome, Kimberly, and thank you so much for being with us today. Hearing your voice and representing the patient voice is extremely valuable and critical to this process. Thank you. Okay, and next we have Dr. Adnan Syed.
Hi, good morning, everyone. I'm Adnan Syed. I'm a professor of medicine, GI, and hepatology at the University of Wisconsin School of Medicine and Public Health. And thanks for inviting me. Happy to be here. No update on my conflicts.
Thank you, Dr. Syed. And Dr. Danielle Brandman.
Good morning. Thank you for having me. Danielle Brandman. I'm the medical director of liver transplantation at Weill Cornell Medicine in New York City, and I do not have any updates to my conflicts.
Again, thank you both for being here. Hearing your clinical expertise and perspective is extremely valuable, and we appreciate you being with us today. Okay, I believe with that, back to you, Steve.
Thanks, Reem. And welcome again to our patient expert and our clinical experts, and to those of you listening in. At the beginning of each, I'm Steve Pearson, I'm the president of ICER. At the beginning of each of our meetings, we want to ground it in a certain purpose of why we are here today and what we hope to accomplish. And I'd like to start, as we always do, with a comment from a patient or a caregiver. This comment says, because I do not look physically sick, some just can't believe the seriousness of the disease. The hardest part is coming home after getting certain results and trying to keep a poker face when a recent test came back showing advanced fibrosis. It's not an easy disease to live with. I pray for a cure one day soon. So one of the key reasons we are here today is to address this unmet need for patients who may not have even been diagnosed yet, or the many who have been diagnosed with non-alcoholic steatohepatitis. There are other reasons that we're here today, and it includes the fact that we are meeting before the FDA has formally decided to approve the two treatments that we'll be discussing today. And what happens the day that any treatment for a disease for which there's an unmet need happens, what happens when they're approved is that there's a really welcome sense of celebration, because the fact that there is a new treatment represents years and years of time, of risk, of investment, of money, and of effort, not just by the drug makers and clinical research folks, but also from patients and their families. And the entire community has a sense of celebration that all of this effort has now provided something that at least can potentially help patients and give clinicians new options. At the same time, when FDA approves something, there's still important questions that really kind of bear consideration. First, what are the true risks and benefits for individual patients to the extent that we can determine that at launch? There's often a lot of uncertainty. And in addition, there are sometimes important questions about how these new treatments really fit into the pattern of care, into an evolving landscape of diagnosis, of kind of prioritization of which patients should be treated and which don't need to be. And all of this kind of comes into greater focus when we have a new treatment approved by the FDA. In addition, there are questions about the pricing, because the day that a new drug is approved, the company is at liberty to set its launch price. And obviously it needs to think about the competitive landscape and other factors, but we have a system in which to the current day, there's a lot of freedom given to companies to set that launch price without regulation from the government or others. So there are reasonable questions about what prices and costs will be to patients and to the health system and the government, and whether those prices and costs seem fair. We also just broadly want to think about the lessons that can be learned from the development to the kind of approval of a new treatment and going forward. What can we learn from the experience to guide our actions in the future, both for this condition and for others? Now, these are in a sense, if you will, the obvious questions that we need to address, but there's another important reason we're here today, and that's because along with the focus on the patients with NASH and their families, the impact of rising healthcare costs affects everyone. And we know that in the United States, we still have problems with affordability, and these affect real Americans. There was a survey done last year by Kaiser Family Foundation that turned out that 100 millions, by extrapolation, 100 million Americans have medical debt today. And that means that they are carrying that debt on their credit card, they can't pay it all off yet, or they're already kind of in deep debt and are trying to struggle. And many of them are actually already being hunted by people trying to retrieve the money that they say that they're owed. And this has real effects on people. In this article, several families and individuals were highlighted and their stories were kind of presented. The family on the left, Marcus and Allison Ward, they had premature birth of their twins and racked up enough costs that were not covered by insurance that they had to kind of switch jobs and move to live with family across the country, and really kind of completely disrupted their lives. And they still are in medical debt years later. Sam and Ariane Buck in the middle are kind of an ironic, if you will, American story. He was a salesman selling insurance, Medicare Advantage plans to individuals and couples. When he became ill, and he and his wife ended up not having adequate insurance coverage, and the extended out-of-pocket put them into medical debt, and they've had to declare medical bankruptcy. Similarly, the woman on the far right, Sherry Foy of West Virginia, she was on a plan that, again, had a cap on the total lifetime expenses, and her condition exceeded that, and so she became deeply in debt and had to declare medical bankruptcy. Now, none of these problems are directly related to drug costs or to any particular drug and its costs, but it's just part of this broader system in which we as a society, and every society, not just America, ultimately wrestles with how to try to provide the best healthcare for everyone with somewhat limited resources at the end of the day. And by the way, of that 100 million Americans, 1 in 7 of those, and if you do the math, that's around, right? It's about 15 to 20 million Americans, said that they had been turned away from healthcare because of their medical debt. That they could not go to the doctor's office, they tried or gone to the hospital and been turned away. And that has real health consequences. So we're here today to try to recognize the implicit balance that we need to find in our health system. And that balance really involves seeing the patient and their family at the center of our concern. And we will talk a lot again about what we can learn about NASH and what we can learn about how to treat patients with new options that may be soon available. But as we focus on them, we need to consider the families and individuals who are part of our broader community, for everybody around whom we need to create a kind of an affordable system that still provides good access to innovative care, but at prices that align with the benefits to patients and can manage the overall affordability so that everyone has access to good care. So we're here to try to grapple with that tension and to deal with it with evidence at the center. You've heard introductions from the Midwest Comparative Effectiveness Public Advisory Council, or CEPAC as we call it. Just a few brief words about ICER, which is the convening host of these meetings. We're an independent not-for-profit institute. We keep an updated pie chart of our overall funding on our website. As you can see, 65% for this year is coming from nonprofit foundations, none of which are directly tied to the healthcare industry. The chief funder within that set of foundations is the Laura and John Arnold Foundation, but there are others, the Commonwealth Foundation, the California Healthcare Foundation, and others. We do take in total about 25% of our overall revenue directly from a mixture of health plans, provider groups, and life science companies, but that is kept sequestered, if you will, separated out for the use of what we call our policy summit and evidence-based dialogue webinars for those companies, because we want to try to work with them to look forward and to make sure that we're working proactively to try to understand how we can get better evidence linked to better coverage and fair pricing for everybody. We have a limited amount of money now coming from subscriptions to our ICER Analytics online platform. Always happy to talk more about that at another time, and a small amount from individual philanthropy. All right, so the report that's the substrate for the discussion today started almost 8 months ago with an intense phase of what we call scoping. And that means that we go out and we talk to patients, to patient groups, to clinical experts, to the manufacturers, and to other stakeholders to really try to get oriented around what matters most to patients. What is the arc of innovation in this area? Where are the drugs coming from? Where are they going? What else is coming? What do we need to really learn before we try to dive into the evidence? And once we kind of get that orientation from others, we start our own review of the evidence, published evidence, sometimes evidence that we can get in confidence from the drug makers themselves. And we try to use that and look at it objectively and to create a cost effectiveness model to look at the long-term value. This goes through several different cycles of public comment and revision. And the draft report, we were fortunate to have expert reviewers, including Dr. Saeed, who's with us on the panel today, also Dr. Mina Bansal, as you can see, William Wong and Jeff McIntyre. So how is the evidence structured in the report in a way to kind of support the discussion today? Well, in some ways you can picture that as a view of our value assessment framework, because ultimately what we're doing is trying to look at the different pieces of evidence as it sums up to considerations around the long-term value for money of different healthcare treatment options. And this starts at the base, if you will, with looking at whether new interventions can help us by providing longer life, extending life of patients. Maybe it does that or it doesn't, but it can also include better health benefits by returning people to higher function. We're having fewer side effects than other treatment options. When we look at cost as a part of value, we're not just considering drug costs. Our analysis really looks at the costs across the entire healthcare system, and even the cost effectiveness modeling in one perspective will look at costs outside the healthcare system. So it's not just drugs, it's looking at doctor visits, potential hospital stays or surgeries, and other costs, because we want to think about the value in the long term, looking at broader costs. And then we'll talk and have some votes actually on two other elements that are sometimes more difficult to pin down with quantitative evidence. But these are things that could be considered benefits beyond health for the individual patient. But the benefits that might stretch over years and years of a patient's life. Also benefits that could accrue to their caregivers. And beyond that, also we talk about special priorities, be they social or ethical, that we may want to consider in this context. And this could be looking at perhaps the severity of the condition, whether there are other treatments available, and some of the other kind of special ethical issues that we might need to bring to the table. So the report has sections on all of these elements. The discussion and voting today will cover these elements. And so I'm looking forward to the conversation with you all. The agenda will start with presentations of the clinical and economic evidence with discussion. The patient representative and our clinical experts will be fully available to the CEPAC during that time. We're going to have a public comment and discussion session that should begin approximately 11:40, followed by a brief lunch break, and we'll return to have further deliberation and the votes, as I mentioned. And then I want to also highlight that we'll be having a policy roundtable discussion starting at 2 p.m. Central. And that's where we'll try to, if you will, take the evidence as it's been viewed and discussed and voted on and look forward. How will that evidence inform coverage policy by insurers? What lessons does it have to tell us about the pricing and how much patients will be paying out of pocket? What do we need to talk about in terms of future research and how to make sure that the clinical application of this evidence meets the best outcomes that we can achieve? So we'll be done by 4 o'clock Central after this forum of discussion, and we'll finish it up with reflections from both the policy roundtable members themselves and the Midwest CEPAC. So with that, let me hand it over to Dr. Jeff Tice, Professor of Medicine at UCSF, who's going to start with the presentation of the clinical evidence. Jeff?
Great. Thanks, Steve. Good morning, everyone. Thanks for your participation. Next slide, please. So I just want to acknowledge initially my collaborators in developing the evidence section for the report, Shahryar Fahim and Belen Hagiwara at ICER and Dr. Janet Chu at UCSF. The only one with a conflict report relevant for this report is Janet Chu. She has received research support from Gilead on identifying NAFLD and NASH in primary care clinics. Otherwise, we have no disclosures to report. Next slide, please. So as we're all aware, non-alcoholic steatohepatitis is common. The sort of background fatty liver disease is present in almost 1 in 4 US adults. NASH itself, so NAFLD with fibrosis is somewhat less common, but still has a very high prevalence, somewhere between 2 and 6% of US adults. The risk factors, primary risk factors for NASH include obesity, diabetes, and the metabolic syndrome with lots of overlap. Important for the discussion today is that NASH is a silent disease until very late in its natural history. Most patients are asymptomatic until they are close to cirrhosis or have cirrhosis. And even though it's asymptomatic for a long time, it can cause severe disease, as we've already heard from the one patient. It can progress to liver failure requiring liver transplantation and all of the many complications that go along with cirrhosis. Next slide, please. So the real impacts on cirrhosis of this disease are of NASH on patients is cirrhosis that patients often report significant fatigue and cognitive issues, often described as brain fog. And the medical issues are decompensated cirrhosis, including the development of ascites, fluid in the abdominal cavity, encephalopathy, and dilated veins, varices, particularly in the esophagus, which can bleed catastrophically. There's also the development of cancer of the liver, hepatocellular carcinoma, and often the only option for managing this illness is liver transplantation, but many patients unfortunately do die from the disease. Next slide, please. The current standard of care is really lifestyle changes focused on weight loss, so exercise and diet. And we all know how challenging it is to make these changes and stick with them for the long term. Bariatric surgery does work for some patients, but it's an uncommon and, of course, a very challenging approach to treatment. The other important part of the standard of care is treating risk factors for cardiovascular disease. So treating their underlying diabetes, treating the dyslipidemia, treating their hypertension. And that's because the #1 cause of death is not liver disease, but actually cardiovascular disease because of the overlap between the risk factors for cardiovascular disease and the risk factors for NASH. And currently, there are no FDA-approved therapies for NASH. There are a few therapies that have been studied and are sometimes used, vitamin E and pioglitazone in particular, but they're not approved, and they've been falling out of favor with clinicians and with patients. Next slide, please. To focus the review, both drugs are really focused right now on NASH, adults with NASH, with stage 2 or 3 fibrosis. So no fibrosis or very mild fibrosis are stages 1 and 2. Stage 4 is cirrhosis. And when patients reach cirrhosis, we think that's already too late. So we really want to attack the disease relatively early. And right now, the focus of the studies has been on patients with stage 2 or 3 fibrosis. Next slide, please. The 2 interventions that we're going to consider today are resmetirom. It's been studied at 2 doses in the pivotal randomized trial, 80 or 100 milligrams. It's a pill taken once a day. and it is a thyroid hormone receptor beta agonist that acts primarily in the liver. The second agent is obeticholic acid. It's also a pill, 25 milligrams taken by mouth once a day. Initially, a 10 milligram dose was also studied in the pivotal trial, but was not effective, and the focus has really been on the 25 milligram dose. It's a bile acid analog approved for the treatment of primary biliary cholangitis. So our clinical experts likely have some experience with the disease in patients with this serious liver illness. But now it's also being studied for a new indication, NASH. Next slide, please. Primary outcomes are intermediate outcomes. The FDA will consider approval based on improvements in these two outcomes, and they are the co-primary outcomes of the primary clinical trials. The first is at least a one-stage improvement in that fibrosis, so going from, say, F3 to F2 without worsening of the markers of NASH on a scale called NAS. Or resolution of NASH without any worsening of fibrosis. So those were the two co-primary outcomes in the clinical trials that the FDA set as the standard for companies. However, the outcomes that matter to patients, as we've already heard, are cirrhosis and the complications of cirrhosis, quality of life, and preventing death from this disease. Next slide, please. We spoke, as Steve mentioned, extensively with patients, and there were a number of themes that I just wanted to highlight here. First, that patients feel frustrated with the lack of knowledge about NASH among both doctors and the public. They expressed a lot of frustration with the intense lifestyle interventions that are required to achieve and maintain weight loss, both the availability of these interventions, the lack of insurance coverage for kind of the level of intensity required to do this, and just the challenges of maintaining that. They also talked about the stigma of the diagnosis of cirrhosis and as many patients are not identified until they reach cirrhosis. The assumption often in their interactions with the public and sometimes with the medical system is that cirrhosis is due to alcoholism, and there's a lot of stigma around cirrhosis. In patients who do have to have a liver transplant, they wanted to highlight just the burdens associated with living post-transplant, the costs and side effects of the medicines they have to take every day to prevent rejection, and that just getting a liver transplant doesn't really mean they return to a normal life. Finally, there was a tremendous amount of financial strain with the illness and treatment. It places a lot of burden on caregivers as the disease progresses for the patients. And they expressed hope that finally we would have an FDA-covered therapy, and that would change the course of disease for themselves, their loved ones, and other patients. Thank you. Next slide, please. So with that, I'm going to turn to the clinical evidence. Next slide, please. I'm going to focus primarily on the phase 3 trials for resmetirom. It's the Maestro-NASH trial. Patients were randomized to either placebo or the 80 or 100 milligram dose of resmetirom, a little over 300 patients in each arm. And the follow-up for the current outcomes that are available were 12 months. The primary outcomes, as discussed earlier, were two, a decrease in the fibrosis stage with no worsening of NASH, or a NASH resolution with no increase in fibrosis. For obeticholic acid, the primary randomized trial is the Regenerate. It was actually a 3-arm study, as I noted before, but we're focusing on just a placebo and 25 milligram dose of OCA. There were a little over 800 patients in each arm. The initial results were presented at 18 months. And I will highlight here that both trials are ongoing. The FDA is requiring long-term follow-up for both total mortality and liver-related outcomes. So we will have data on clinical outcomes that matter, but the estimates are that that's going to take somewhere between 4.5 and 7 years of follow-up with these trials. Next slide, please. So I'm going to discuss the results of resmetirom first. Next slide, please. So here are the primary outcomes. In the improvement in fibrosis with no worsening in NASH, that occurred in 14% of patients in the placebo group, highlighting the importance of having a placebo group here because you do have regression of fibrosis, at least by biopsy, in patients receiving no specific therapy other than the background lifestyle interventions, which all patients receive. In the resmetirom group at 80 milligrams, a higher percentage had regression of fibrosis, 24%, and slightly higher even in the higher dose at 26%. Both of these were statistically significant compared to placebo. Similarly, for the outcome of NASH resolution without worsening of fibrosis, 10% in the placebo group, 26% in the 80 milligram resmetirom group, and 30% in the 100 milligram resmetirom group. Again, highly significant, statistically significant differences. Next slide, please. There's been a lot of interest in lipids. In fact, resmetirom was initially developed as a therapy to treat elevated cholesterol, to lower LDL cholesterol. And indeed, in the MAESTRO-NASH trial, there was a 12% reduction in LDL cholesterol in the 80 milligram group, 16% reduction in the 100 milligram group versus a slight increase, a 1% increase in LDL cholesterol in the placebo group. These were highly significant again, but these are, again, intermediate markers. We don't know whether that translates into a reduction in cardiovascular events long-term, but that would be very important if it did. I want to highlight here that currently there are no reported results on quality of life, fatigue, or functional status from the clinical trial. All we have are top-line results reported by the company, no detailed publications on the outcomes yet. Next slide, please. For harms, serious adverse events occurred in about 12% of patients in all three groups, so no important differences. There were clearly an increase in diarrhea and patients having diarrhea in the resmetirom groups, 28 and 34% of patients in the active treatment groups versus 16% in the placebo group. And discontinuations due to adverse events weren't really different between the placebo and 80 milligram resmetirom dose, but appeared to be higher in the 100 milligram dose, so 7.7% versus 3 to 4% in the other two groups. Next slide, please. So that's the clinical evidence for resmetirom. I'm going to turn to obeticholic acid now. Next slide, please. Again, the first, the two outcomes mandated by the FDA. The first reduction in fibrosis occurred in 9.6% in patients in the placebo group, 22% in the obeticholic acid group, a highly statistically significant difference. And NASH resolution occurred in 3.5% of the placebo group, 6.5% in the OCA group. This difference was not statistically significant, but was moving in the right direction. Next slide, please. For lipid outcomes, this is a concern with obeticholic acid. At 1 month, there was actually a 24% increase in LDL cholesterol levels compared to a 3% reduction in the placebo group. However, by 18 months, these differences were no longer statistically significant. However, it's important to put that in the context of intensification or new statin therapy. So 159 patients in the obeticholic acid group started statins for the first time during that follow-up period versus 66 patients, in the placebo group. So part of the reduction in the difference in LDL cholesterol was due to the starting of cholesterol lowering medications. We also don't know about intensification of therapy in patients already on statins at the start of the trial. They did report quality of life outcomes using a standard measure, the EuroQol-5D, and there were no differences overall or on any of the subscales at any time point. This was assessed multiple times through the first 18 months of the study. Next slide, please. In terms of harms, serious adverse events were nominally higher in the OCA group at 26% versus 22% in the placebo group. The most common adverse event was pruritus, itching, in about 55% of the obeticholic acid group versus 24% of the placebo group. And this and other adverse events led to a higher rate of discontinuation of therapy, about 22% versus 11% in the placebo group. Next slide, please. So there are a number of controversies and uncertainties. I think the most important one is that NASH is a slowly progressive disease, and the current clinical trial results really just present short-term outcomes on intermediate markers of disease. We don't yet know whether these will translate into reduction in the incidence of cirrhosis and all of the complications of cirrhosis. I highlighted the cholesterol results again because cardiovascular disease is the #1 killer in patients with NASH, and OCA initially raised HDL, though that came down with intensification or starting of lipid-lowering therapy. Resmetirom, on the other hand, lowers LDL cholesterol. But again, these are surrogate markers. Most of the time, changes in LDL cholesterol result in changes in cardiovascular outcomes, but we have examples of drugs that lower LDL cholesterol and actually increase cardiovascular events. So it's not a perfect surrogate marker. And then finally, we know that in patients treated with OCA for primary biliary cholangitis at doses lower than those studied for NASH at 10 milligrams, 5 or 10 milligram doses, they actually had reports of hepatic decompensation and death in patients with cirrhosis. So there are concerns about severe adverse events from OCA that also give us some pause. It's unclear. This hasn't been reported yet in the studies of NASH. So it may be specific to PBC, but we need to be sensitive to this as a concern. Next slide, please. There are a number of potential other benefits and contextual considerations. One of the complex areas is health equity. Overall, the Hispanic population has a higher prevalence of NASH than the non-Hispanic white population, and the non-Hispanic Black population has a lower prevalence. But when you look at non-Hispanic whites versus all other race ethnicity subgroups, they actually have a higher prevalence. So it's kind of a mixed picture, and we don't know the details really in the population of interest for today, those with stage 2 or 3 fibrosis, but likely similar disparities exist. Another thing that we heard and thought was important in terms of disparities is disparities by socioeconomic status. It's much more challenging for patients without financial resources to qualify for and receive liver transplants. They often have to travel long ways. They have to demonstrate that they have the kind of support needed to guide them through receiving the transplant and the challenges of post-transplant medications. And there is a potential that access to these new therapies could reduce this disparity. Next slide, please. We had a number of public comments. The most common one was that the health equity section was not fully developed. I just highlighted some things that we have expanded in the report. We also heard that the reduction in the need for liver transplantation for patients with NASH, if these drugs are effective, then could increase the supply of livers available for patients with other diseases who require liver transplantation. Next slide, please. So in summary, for resmetirom in patients with NASH and fibrosis, so F2, F3 fibrosis, resmetirom appears to reduce progression and promote regression of fibrosis, and thus lead to a resolution of NASH compared with placebo, again, in top-line unpublished results. There are uncertainties about the long-term benefit and magnitude of these changes. The harms appear small, though diarrhea is common, and there were more discontinuations due to adverse events in the high-dose resmetirom group. Finally, LDL cholesterol levels were lower, but it is unclear whether this will translate into a reduction in cardiovascular events and death over time. Next slide, please. So our evidence rating for resmetirom is C++. That means we feel there's moderate certainty of at least comparable, so no worse than standard therapy, but the possibility of significant or substantial net health benefits. But because of the unpublished nature of the data, the fact that they're mainly based on intermediate outcomes, we feel like there's substantial uncertainty about what the actual net health benefits will be. Next slide, please. For obeticholic acid, again, OCA appears to promote regression of fibrosis compared with placebo. There's uncertainty about the long-term magnitude and impact of these changes. It certainly causes pruritus and so may worsen quality of life in patients who were previously asymptomatic. And again, when used for primary biliary cholangitis, sorry, that's a typo there, there have been reports of severe harms with liver decompensation and death, and our increase in LDL cholesterol is also worrisome. So considering all of the evidence, we feel like the long-term net health benefits of OCA on quality of life and health and benefits are uncertain. Next slide, please. So that results in a promising but inconclusive result, meaning that there could be, we still think there's the possibility of net harm. We really don't feel like there's sufficient data to give a more positive evidence rating. Next slide, please. And with that, I can open it up for questions.
Well, thank you so much, Dr. Theis, for this excellent summary. For two different medications, highlighting what the studies show, but also some of the uncertainties. With that, I'm going to ask the CEPAC members, please raise your hand if you have any questions. And I'm just going to kind of explicitly state, we are quite excited that we have a lot of public commenters today, but that does mean a challenge with time management. So part of my role is to make sure we stay on time. So I ask all of you, please raise your hand, ask your questions. And if we have to cut some of this discussion short, I do apologize. Okay, Dr. Tim Welt.
Hi, thanks. And again, really a great presentation. I still struggle to find how you came with a C++ for clinical benefits when all of the markers there are putative surrogates. And as you stated, long time for follow-up, and there's actually some harms with that. You state that they're going to be long-term follow-up 4 to 8 years, but do you really think that that's sufficient follow-up time given some of the that these are stage 2, stage 3, not even cirrhotic patients yet, and that being based really on studies of modest sample size? So maybe just, so.
Important comments. Thank you, Dr. Wiltz. I believe both studies were powered for clinical outcomes based on estimates of transition from F2, F3 to F3 and on to F4. So there is hope for that. Sometimes, as you're well aware, patients that agree to participate in clinical trials are sometimes healthier and maybe are more adherent to lifestyle changes, and maybe they will progress more slowly and there may need to be longer follow-up. We'll have to see. My understanding is that the studies were powered for those clinical outcomes in the long term, and perhaps our clinical experts may know more than me on this.
But before you get to that, I just wanted to jump in and remind Dr. Wilk that the C part of the C++ is basically just a statement that we think it's unlikely that the drug will be worse than placebo. And so we're suggesting that there's a very wide range of uncertainty there. The conclusion that the drug is unlikely to be worse than placebo is because of its LDL lowering effects. We had some discussions about whether we could even go that far with it. So, if you came to the conclusion that you still worried that it was worse than placebo, it's not an unreasonable position, but our group position belief was that any other harms it had were unlikely to offset the improvement in lipids.
Thank you, Dr. Wren. Okay, Dr. Kerfman.
Thank you very much. And thank you very much, Dr. Tice, for a really interesting presentation. Dr. Wilt has just pointed out and underscored the fact that we're dealing here with surrogate endpoints and not clinical endpoints. We do have a study involving bariatric surgery from the Cleveland Clinic that does have clinical endpoints, published in JAMA in 2021. It was not a clinical trial, but a retrospective cohort study, but very carefully matched all patients with NASH matched to bariatric surgery or standard therapy, with really a very marked reduction in serious hepatic outcomes in the patients undergoing bariatric surgery. It's just something to keep in mind. I'm not suggesting that bariatric surgery would be necessarily a common therapy for NASH, but as has already been alluded to, we are on the cusp of having some real significant oral therapies for obesity and overweight. And so I think we have to keep in mind that fibrosis in the liver is related to fat in the liver, and the most direct way of dealing with the problem is going to be to remove the fat. Thank you.
Yeah, thank you. I think there was just either a randomized trial to either Roux-en-Y, gastric bypass, sleeve gastrectomy, or medical therapy that some initial results were just released that were promising as well, though not yet with clinical outcomes. So certainly that's why I highlighted bariatric surgery, and I think many people are excited with the potential for the GLP-1 or the dual receptor agonists that are coming down the pike for weight loss equivalent to or close to that with bariatric surgery. We'll see whether that plays out as an effective treatment for NASH. They're being studied. We just don't have data yet.
Thank you. Excellent comments. Also keeping in for all of us the big picture about different options. Okay, Dr. Armbrecht.
I think Dr. Rind answered my initial question. So, but I was curious about I view the sort of main endpoint here as transplant prevention. Is that the right way to look at this? And we just don't have that data. And so I have to put faith in the surrogate markers, which I found interesting in the placebo group were they moved considerably just with, kind of routine treatment. So I found that gave me pause to think about the effectiveness. So that's how I'm thinking about this. So the effectiveness of the treatment, given that a lifestyle intervention had such a remarkable effect in the placebo group, which presumably also would extend the number of years that someone would be transplant free. And I'm kind of asking to the evidence team, like, am I thinking about this the right way, or did I misunderstand something along the way?
No, you understood it correctly, but I think there are concerns, and maybe our experts want to comment on this about sampling bias. We're just sampling a small percentage of the liver. It could be there are various stages of fibrosis throughout the liver. It may not be uniform. And so this may not be the perfect, even liver biopsy may not be or change in liver biopsy may not be the perfect outcome. It may be a sampling issue.
I see Dr. Saeed's hands up. I believe you want to comment on that.
Yeah, no, I think those are some really valid and excellent points, because we are using a surrogate marker, and there is no direct evidence, but the studies are going to collect the clinical outcomes you're looking at. So that's decompensated cirrhosis or development of cirrhosis, because we know cirrhosis has a adverse impact on life, on duration of life, as well as quality of life, right? We were talking about the brain fog and the risk of developing cancer. So those outcomes should come. This is not unique to NASH, but this is a concern in a lot of chronic diseases. How does surrogate marker improvement lead to clinical improvement? And in just the liver arena, you can look at the example of PBC, where improvement in alkaline phosphatase leads to improvement in clinical outcomes and cirrhosis. And so the FDA allows that as a surrogate marker. Hepatitis C has been a great success story. We didn't have a lot of great evidence that getting rid of the virus makes a clinical outcome, but it just makes sense because you're completely getting rid of the disease. And we are seeing that evidence now play out years later. But there is a strong component of lifestyle modification here as well. And in these studies, there was a decent placebo impact, even without intensive lifestyle modification. So the control arm was lifestyle modification, but it wasn't intensive lifestyle modification. So there is no comparison with that group that we were discussing, right? The intensive nutrition and weight loss therapies versus these drugs. Are they equivalent? Are the drugs still going to be better? Is there a phenotype? So those are important questions, and hopefully the clinical answers will come in several years, not immediately.
Thank you, Dr. Saeed. And Dr. Brenman, I see your hands up, and I'm assuming you want to comment on the same point. So I would like you to comment, and then we'll go to Dr. Huang.
Great, thank you. Yeah, and just to speak to the point of the surrogate marker, and just thinking about how does NASH actually progress and how long does it take to develop events? We know that after about 10 years of follow-up in patients with cirrhosis, almost 20% will experience an episode of decompensation, or basically sign of liver failure. And then, almost 10% may develop liver cancer. So, I think in order to see those outcomes, this extended follow-up beyond the standard clinical trial assessment of the histologic endpoints, I think is really important. I don't think it's necessary for drug approval yet, because I think if we wait for that time, I think it would be really challenging in order for us to find drugs that are effective, just because you have to follow so many patients for such a long period of time. And then just to talk a little bit about the lifestyle modification, is that we know that our patients have so many challenges in adhering to lifestyle modification, and fewer than 10% reach the threshold of 10% body weight loss that is needed to improve liver fibrosis. So I think it's great we are seeing some improvement in histologic markers in the placebo patients, but in order to really be successful, there have to be, I think, a lot of resources dedicated to promote sustained weight loss in those patients.
Excellent. Thank you again, highlighting the challenges with some of the other potential interventions. Dr. Albert Huang, I would like to ask you to introduce yourself and tell us if you have any conflicts, and then you can ask your question.
Great. Yes. Hi, I'm Albert Huang. I'm a professor of medicine and public health sciences at the University of Chicago. I'm also a practicing primary care doctor, which is why I was delayed this morning. My apologies. And I do not have any conflicts. So my question is actually related to the degree to which we can have confidence in extrapolating intermediate outcomes to long-term outcomes. And this is very similar to other areas like chronic kidney disease, diabetes, hypertension. And so I just wanted to know if the experts or Dr. Theis could talk about the degree to which there's confidence in the epidemiology of NASH Because, in many areas, even areas like HIV, we made policy decisions based on forecasting models, simulation. They're all based on the epidemiology of the disease. And so if we have confidence in the epidemiology of disease, then I feel better about making the leap about, interpreting the evidence from the trials.
I'm going to let the experts weigh in because I think that would be more time efficient.
Yeah, I mean, I think we know that fibrosis is the most important predictor of outcomes in these patients. And yes, it's overall mortality, but also liver-related outcomes such as liver failure, liver cancer, need for liver transplant, and liver-related death. So I think if we're looking at fibrosis as a surrogate outcome, with the expectation that if you improve fibrosis, I would expect that you're going to reduce the incidence of those liver-related events.
I would endorse that. There is moderate degree of evidence that fibrosis would lead to progression and lead to these bad outcomes. From prospective studies too, even over a short period of time, the NASH Clinical Research Network, in a relatively short period, less than 5 years, between 4 and 5 years, showed that F3 fibrosis is associated with worse outcomes, even in that short period, in terms of cirrhosis, liver cancer, and complications of cirrhosis and death that we were talking about. Not shown clearly for F2 yet, so that's more of an extrapolation. So we have more evidence or weight of evidence that F3 is bad. Not as bad as F4, which is cirrhosis, but definitely worse than F0 to F2. F0 to F2 is kind of bunched together, but I think it's more of an extrapolation, but I think that also will lead to the same progression, not quite as fast and much slower. So I agree with that.
And I wanted to highlight one of the most, there's consensus among the patient community that even just halting the progression, so just staying at the same stage would be a huge step forward for this. So they would be very happy. And essentially in both of those intermediate outcomes, you had no increase in fibrosis stage. So it certainly is, these are outcomes that patients feel passionate about.
Excellent. Well, thank you for this, all these really important questions. Thank you, Dr. Tice and our clinical experts for addressing them really in a very thoughtful way. I'm actually going to ask to move forward to our next part, and I would like to introduce Dr. Kang-Hoo Soo to present about the evidence and economic modeling.
Hello, everyone. My name is Kang-Hoo Soo. I'm an assistant professor at the School of Pharmacy at University of Pittsburgh, and I'll be presenting the economic evaluation section of this analysis. Next slide. I would like to thank some key review team members, Dr. Josh Carlson from the University of Washington, Ronald Dickerson, also from the University of Washington, and Marina Richardson, senior health economist at ICER. Dr. Carlson and I have no conflicts to disclose. So the objective of this section is to evaluate the lifetime cost effectiveness of resmetirom or obeticholic acid compared to standard care for the treatment of NASH. And I'll be discussing these methods in brief. Here are the methods that we use. These are standard Markov model methods used in previous ICER reports. Of note, I would like to highlight that we used a 1-year cycle length, and the primary outcomes that we looked at were cost per quality adjusted life year, cost per life year gained, and cost per equal value of life years gained. Next slide. This is the model schematic that we used. We have two submodels. One where the first model where there's no prior cardiovascular event, and then the second model where the patient could have experienced a prior cardiovascular event. And within these two submodels, patients could transition between fibrosis stages with NASH and then they could progress to more advanced liver health states, such as decompensated cirrhosis and liver transplant. Next slide. In terms of the model characteristics, we had patients start the models in NASH fibrosis stage 2 or 3. We modeled 2 separate NASH populations. The first was resmetirom versus standard care. And then in our second comparison, we assessed obeticholic acid versus standard care. Next slide. We had the full list of our model assumptions are in the final report, but here we would like to highlight a few of them. The first is that the treatment effects for improvement and worsening were used as the basis for deriving transition probabilities among fibrosis stages. And these were applied uniformly regardless of starting stage. The second key model assumption is that in the absence of detailed resmetirom phase 3 trial data, we assume that the composite of improvement in fibrosis without worsening of the NAFLD activity score between treatment groups was comparable to improvement in fibrosis alone. Third, patients who entered the prior CV event submodel had the same per event cost, quality of life, and mortality regardless of subsequent CV events. And this is a byproduct and limitation of using Markov models. And finally, lipid levels have a direct impact on CV events and mortality in our model, although this has not been shown yet in clinical trials. These are the efficacy parameter inputs that we used. I would like to first highlight the standard care probabilities. These were taken from the Regenerate trial. And so from that trial, we saw an improvement of fibrosis of 23% and a worsening of 21%. And from these baseline standard care probabilities, we applied absolute risk differences for resmetirom and obeticholic acid based on based on their respective phase 3 results. Next slide. We needed further information to inform fibrosis transition probabilities. So if we look at the first green circle to the left, in this example, we knew that patients who were on resmetirom who started in the F2 stage had a 35% chance of improvement. However, we needed more information to inform what stage they would improve to from F2. And so to inform that transition, we used a meta-analysis of previous NASH clinical trials. And from that meta-analysis, we knew that among those patients who improved from F2, 23% improved to F0 and 77% improved to F1. And then we further adjusted these probabilities to fit our yearly cycle in our model. And then that led us to our final stage specific transition probabilities. And so in this example, while we only showed it for F2, we repeated this exercise for all fibrosis stages. Next slide. For drug costs, these are the costs that we used. For resmetirom, we assumed a placeholder price of $19,000 based on an early economic model that was published by the manufacturer. But again, this was a placeholder price. For obeticholic acid, we also used a placeholder price based on other dosage forms that are available currently for another indication. Next slide. Here are the healthcare sector related costs that we used. As you can see, as patients progress from earlier fibrosis stages to more advanced liver health states, the costs increase dramatically. And then in the next part of the table, these are the cardiovascular related events, the costs related to cardiovascular events. And then as you can see, the costs for MI and stroke are also pretty costly as well. These are the utilities that we used for in our model. The NASH fibrosis stage 0 to 2 and 3 values were taken from adult patients with NASH who provided their quality of life using the EQ5D. And we also highlight here the utility values for those in downstream advanced liver health states. And as you can see, the key point here is that the utilities do decrease pretty dramatically from the early fibrosis stages to the more advanced liver states. The next part of the table down below shows the disutilities that we used for cardiovascular events. And these were applied to one cycle, one patient's experienced the event. Next slide. So here are the results for resmetirom, the base case results. You can see that there was an increase in drug costs compared to standard care. When we look at the non-drug costs, we can see that there was a decrease in costs for resmetirom, and this was due to avoiding downstream advanced liver disease events. And so in total, we can see that there was a decrease in total cost for resmetirom of about $22,000. When we looked at the life years qualities and EVLYs, we saw that there was a gain in these outcomes with resmetirom. When we put these two components together and looked at the incremental cost effectiveness ratios, we saw that compared to standard care, resmetirom was less costly and more effective when we looked at cost per quality gained and cost per EVLY gained. Next slide. These are the base case results for obeticholic acid. Again, we see an increase in drug cost with treatment with obeticholic acid. And similarly, we see a decrease in non-drug costs as patients on obeticholic acid avoided downstream advanced liver disease health states. In total, obeticholic acid had an increased cost of about $240,000, but treatment with obeticholic acid also had gains of life years qualities and EVLYs compared to standard care. When we put these two components together and looked at the cost effectiveness ratios, we saw that obeticholic acid had a cost per quality gained of about $560,000 and cost per EVLY gained of about $500,000. This is the sensitivity analyses for resmetirom. The top part of the slide shows the tornado diagram for resmetirom. The most influential parameters were the placeholder price for resmetirom, as well as some utility values. When we looked at the probabilistic sensitivity analyses, using the placeholder price to calculate the proportion of simulations where resmetirom were cost effective across commonly cited thresholds, we found that in these simulations, it was cost effective 99.4% to 99.8% of the time. This slide shows the sensitivity analyses for obeticholic acid. Similarly here, the most influential parameters in the tornado diagram were utility inputs as well as the placeholder price for obeticholic acid. The probabilistic sensitivity analyses showed that the proportion of simulations where obeticholic acid were cost effective across commonly cited thresholds was 0%. Here are scenario analyses that we wanted to highlight. We have a few more scenario analyses in the final report, but here we highlight a few of them. None of them really changed our conclusion about either drug. The first one that we wanted to highlight was the modified societal perspective. The second scenario analysis is an early versus late discontinuation for resmetirom. And this came about because the manufacturer submitted a comment saying that we should be using, instead of using a constant discontinuation probability throughout the model, we should separate that parameter into an early versus late discontinuation probability, but it did not really change our conclusion about the drug. And then in the last scenario analyses here, we assumed no LDL benefit for resmetirom, but in this case, using the placeholder price, it was still less costly and more effective than standard care. Here we highlight the health benefit price benchmarks. In the second column, we show our placeholder prices that we used, about $19,000 for resmetirom and about $85,000 for obeticholic acid. Assuming or across commonly cited thresholds of about $100,000 to $150,000 across QALYs and EVLYs, resmetirom would have a health benefit price benchmark of about $40,000 to $50,000, and resmetirom would have a health benefit price benchmark of about $33,000 to $41,000. We do not show the discount from the placeholder prices to reach the threshold prices because, as mentioned before, the annual prices currently are based off of placeholder prices. Next slide. So we highlight here a few limitations of our model. We did not have detailed data on stage-specific transitions and treatment effectiveness. We did ask for this data from the manufacturers, but we did not receive them. For resmetirom, the model was based off of top-line phase 3 results, and it used a placeholder price. For OCA, the model was based off per protocol estimates from the Regenerate trial and used placeholder price. Next slide. So here are a few comments that we received. So the first was around discontinuation rates, which I mentioned and detailed a couple slides earlier. Another comment that we received was to use non-invasive testing for the current and future NASH models. For the current model, the efficacy parameters were based off of the clinical trial inputs, which were based off of liver biopsy. And so it was a little, so the use of non-invasive testing was a little out of scope for the current NASH model. And finally, we received comments that we should to suggest use of updated costs for NASH health state, for the NASH health state costs. And so we did do that, and we did update the costs. So in conclusion, resmetirom and obeticholic acid provided clinical benefit in terms of gains in QALYs, life years, and EVLYs over their respective standard care alone. At the currently assumed placeholder price, resmetirom would be less costly and more effective than standard care. And at the currently assumed placeholder price, obeticholic acid would not meet commonly cited cost effectiveness thresholds. Thank you, and I'll take any questions. Reem?
Yeah, thank you so much. Again, really nicely done, summarizing a lot of data and highlighting some of the uncertainties. Maybe it's a question, maybe more it's a comment I have as my colleagues are thinking about raising their hands and asking their questions. It was interesting to me to see that the utilities or disutilities of stages F, really 1 to 3, were quite compatible to the post-liver transplant disutilities. That was so interesting. So in my mind, trying to translate some methods into clinical and patient experiences, and maybe Kimberly, I don't want to put you on the spot, but I would love to hear from your perspective. You know, what we're trying to achieve for people who progress is at best to kind of bring them back to what they would have been kind of feeling from a disutility perspective as a stage. And interesting that the disutility for stage 1, 2, and 3 were quite close, actually, not much different. So don't know, Kimberly.
Very interesting. The one thing that I took from it was the cost of the liver transplant. Can you tell me where I could get one for $232,000? I mean, I don't understand. Is that the actual cost before the hospital marks it up, or I don't understand. Is that the up-to-date price for a liver transplant?
Sure. So I'll speak to Kimberly's comment first and then come back to Reem's comment. So with the liver transplant cost, we did receive actually a comment saying that we should have used a higher price. And But the price suggested that we use was based on charge data, which is not the actual cost. And so our cost estimate that we used was from a administrative claims-based study. And so that's the estimate that we found from that published study that looked at NAFLD or NASH patients who actually received a liver transplant.
Okay.
And then and for Reem's comment, so the sources that we did use for the F0 to F3 compared to the post-liver transplant are different sources. So that could be one reason for the similarity in terms of the utility values. I do want to note that for the again, we're using placeholder prices, but when we looked at the one-way sensitivity analyses and the probabilistic sensitivity analyses, where we vary the utility values, our conclusions about either drug did not change.
Thank you very much. That's quite helpful. Kurt, I see your hands up.
Yes, thanks. So just a clarifying question. The resmetirom was used for 80 milligrams, right? So this economic model would not apply to the 100 milligrams. Should that get approved? Is that correct?
It would not, but if the 100 milligram dosage form was approved, we could obtain those estimates, the clinical effectiveness estimates, as well as if there is a different price for it, and we could input those values into the model to see what the result would be.
Okay, thanks. And higher discontinuation rates, obviously, as well.
Right, yes, and discontinuation rates
you.
could also be, customized.
Excellent. Okay, Tim Well.
Thank you. And again, you guys really present this cost stuff very clearly and nicely. So thank you. Regarding the disutilities, et cetera, for 0, 2, and 3, building on Reem's comment, given that we heard from our experts that those individuals are almost always asymptomatic, et cetera, I would view that almost as just a base state of their underlying health unrelated to their liver disease. I may be wrong, but be interested to hear that. And then because so much of this is dependent on our certainty of the stages and changes in stages leading to real health outcomes, symptoms, symptomatic cirrhosis, decompensation, liver cancer, need for transplant. How sensitive were your results and how certain are you about the results, given that relatively short-term data from the studies and inputs from other kind of assumptions, and whether the patients in these trials are really representative of who the clinicians are going to really see?
Sure. So for the disutilities, those were taken from a study that assessed adult patients in the U.S., but not specifically NASH patients. So if disutilities were studied in NASH patients specifically, we could find different results. But so I'm not sure if that really addresses that question. But for your second question, I think based on what the clinical experts have said about fibrosis being the key driver of progression in NASH, where we are basing our model based off of fibrosis. And so I think if that is true, then we can be fairly confident in our model results. Additionally, that is why we also run sensitivity analyses where we vary one parameter at a time, and then also where we vary all of them together at the same time simultaneously. And in both scenarios at the placeholder price, again, we found consistent results. And so I think we are fairly confident in our modeling.
Okay. Ingve, I see your hands up.
Just real quick, because it has been now mentioned with the disutilities or the utilities at baseline at 0 to 2 being 0.76 being a little bit aggressive. Can you just comment if we would increase this to a more reasonable, something like 0.9 or something, that would actually strengthen your cost effectiveness, correct? It wouldn't really dilute it down. Is that correct?
Let me
because usually when you go from a higher state and you can prevent a lower state, what would happen is it would make it more cost effective.
Right.
But I just want to confirm that.
So we actually did, I believe, give me 1 second. Trying to get to my slide.
And while you do that, I think I have to agree with you that actually the fibrosis state has a very rich literature in terms of progression over time from indirect sources as well. So we know actually quite fairly well what happens over time when you have inflammation in terms of fibrosis stages. There are differences in velocity, there's no question about it, but I believe that the way you did it is the only way we can do it with NASH. Since doing clinical studies for 20 years, it's going to be really hard to do.
Yeah, so for the fibrosis stage 0 to 2, when we did the one-way sensitivity analysis, we did use a range from 0.61 to 0.91. And even at the upper range of 0.91, we still found that resmetirom was less costly and more effective. And for OCA, we found, we still found that at commonly cited thresholds, it was not cost effective.
It's expected. Thank you so much.
That's really, really helpful. I think highlighting that despite maybe even some uncertainty around these utilities, I think your results are consistent. And Ingve, I will just explicitly address that we're quite fortunate to have you also as a hepatologist and expert in this field on our panel. So we appreciate your expertise. Thank you. Ellen, I see your hands up.
Thank you. And I'm pretty sure I know the answer to this question, but it's not meant to be rhetorical, but it maybe just make sure we're all on the same page. For the purposes of this analysis, the standard of care comparison was similar to or equal to the standard of care in the trials, right? So in terms of the level of lifestyle management. So we're not comparing this to intensive lifestyle management, but whatever the lifestyle management that was in the standard of care for the trials, correct?
Right, exactly, yeah. So the standard of care that we used was directly from the Regenerate trial, where the comparison was OCA versus standard care, so yes.
Just to clarify, the standard of care is in both groups, right? So just to make sure that people understand, the placebo response rate, right, has to do with weight loss associated with, when people are enrolled in studies, they tend to actually go down towards that because they get leaflets in their hand, they want to lose weight. So once we have that in both arms and once it's blinded and so on, we should factor this out. That should be equally distributed. That should actually then make sure that we come out with a actual effect estimate. And so I would not put too much, into that there's a certain placebo. That's the whole point. We're doing controlled clinical trial that we can get that one sorted out, right?
Right, excellent. I see lots of head nodding, and I think that's a really important point. I'm not seeing any hands up, and I think that's actually not a bad thing. It highlights that we had excellent presentations. If no other questions, I would like to move to the next part. As I mentioned earlier, we do have a large number of public comments. So I'm going to ask the ICER staff maybe to bring up on the screen